Welcome to YouTube channel. So in this we'll talk about the early experiments like some of the experiments are explained to understand the photosynthesis process. So Joseph Priestley in 1770 performed a series of the experiments that revealed the essential role of the air in the growth of the green plants. So he conducted experiments regarding taking mouse, candle, green plants and a jar. So he placed a burning candle in bell jar. Similarly, a mouse would soon suffocate in closed place and it died. So in other thing he placed a burning candle or animal that breathed the air. So when he placed a mint plant in a same bell jar, he found that mouse stay alive and the candle continued to burn. So when he placed it without the plant, the mouse may die. But when he placed it with the plant with mouse, the mouse is stayed alive. So the candle continued to burn. Plants restored to the air whatever breathing plants and animals can be removed. So using the similar setup once in a dark room in sunlight, Zan in their house showed that sunlight is essential for plant process that somehow purifies the air fueled by the burning candles or breathing animals. Elegant experiments with aquatic plants showed that in bright light, small bubbles are formed around the green parts, while in the dark they does not. Later, he identified these bubbles as the part of the oxygen. It only green part of the plant that could release the oxygen, whereas the dark part it doesn't show any bubbles or anything. Another experiment is conducted based on the Julie Swan's switches provide evidence for the production of the glucose. When plants grow, glucose is usually stored as starch. His later study shows that green plants in plants chlorophyll, as we know it now, located in special bodies later called chloroplast within plant cell. He found that green parts in plants is where glucose is made, and the glucose is usually stored as a starch. And another experiment is T.W. Engelmann. He taken the experiment like a prism, which is he split into the special spectral components and illuminated a green algae cladophora placed in suspension of the aerobic bacteria. So the bacteria used to detect the sites of the O2 evolution. He observed that the bacteria accumulated mainly in regions of the blue and light of the spectrum. His first action spectrum of photosynthesis was thus observed. Absorption spectra of chlorophyll A and B. So in 19th century, key features of plant photosynthesis, we know that they use the light energy to make carbohydrates from CO2 and water. So glucose formula is C6H12O6. Empirical formula is CH2O. And another microbiologist to understand the photosynthesis, Cornelius Van Niel. So he studied the purple and green bacteria to demonstrate the photosynthesis light dependent reaction. So these all experiments are regarding the photosynthesis, in which hydrogen from suitable oxidizable compound reduce carbon dioxide to carbohydrates. In green plants, H2O is a hydrogen donor and oxidized to O2. Some organisms do not release O2 during when H2 is instead a hydrogen donor for purple and green sulfur bacteria. The oxidation production is sulfur or sulfate depending on organism. Hence, he is inferred that the O2 evolved by the green comes from H2O, not from CO2. Here the balanced equations are shown that from CO2 and water produces glucose, and H2A and carbon dioxide produces glucose. CO2 C6 CO2 plus 12 H2O gives rise to glucose and oxygen and water. So these all are about the experiments regarding the showing for the understanding of the photosynthesis. We'll discuss later about the where the photosynthesis takes place in the plants.